Good morning. I start today's session by acknowledging the First Peoples, the traditional custodians of the land on which the festival is being presented. At this time, I also recognise and pay my respects to the first scientists of this land. Increasingly, in many scientific fields, we're recognising the important contribution that First Nation science makes to contemporary scientific discovery. Good morning. My name is Aidan Byrne, and I'm the Provost of the University of Queensland. It is my pleasure to welcome you to the Pulse of the Pandemic, a World Science Festival Brisbane 2001 conversation presented in partnership with the University of Queensland. Queensland Museum is proud to be the exclusive host of World Science Festival in the Asia Pacific. Now in its 6th year, World Science Festival Brisbane dares us to imagine what we want our future to be and how science can make it possible. After a year of tremendous upheaval and uncertainty, we turn to art, science, technology and human curiosity to help us understand extreme circumstances and adapt to the new reality. Today's conversation could not be more timely, and I'm sure all of you would have checked today's announcement from the Queensland Government and encourage you all to pursue COVID-safe practices. In a rapidly changing and complex world, Australia's future success is linked to innovation, which requires an educated and diverse workforce founded on strong pipeline of expertise in science, technology, engineering and maths, or STEM. The University of Queensland works in a spirit of genuine partnership with organisations such as the Queensland Museum to foster a society that embraces STEM. And I can think of no better way to achieve this than as an academic partner of the World Science Festival Brisbane, one of the world's most celebrated scientific festivals. The festival would be impossible to deliver without the insistence of many organisations and individuals. And on behalf of the Queensland Museum, I would like to acknowledge all the partnerships that make the festival possible, including the World Science Festival New York team, Queensland Government, Tourism and Events Queensland, Brisbane Economic Development Agency, BHP, Griffith University, QUT and the University of Queensland. Our moderator this morning is multi-award-winning producer and broadcaster, Dr. Norman Swann. Dr. Swann has become a familiar voice of reason in his role as the ABC's health correspondent during the COVID-19 pandemic. He is the host of Radio National's Health Report and co-host of CoronaCast and commentator on ABC TV's 7:30. Before we welcome Dr. Swann, let us hear from two people we have all become very familiar with over the last year. Queensland and Victoria Chief Health Officers sat down with Tegan Taylor to talk about their role in the pandemic. Please enjoy the show.
Hello, I'm Tegan Taylor and I'm here at the office of the Queensland Chief Health Officer, Dr. Jeanette Young, to talk to her and her Victorian counterpart, Professor Brett Sutton, about what it's like doing their jobs, especially over the past year. Jeanette, most people remember March 2020 as sort of the turning point. We were in a pandemic, but is there a date or time that really sticks out to you that this was going to be a world-changing pandemic?
It was 3rd of January when we first heard from the Chief Medical Officer. So that's when I first heard there might be a problem. And then over days, that rapidly escalated.
What about you, Brett? Was it as early as the 3rd of January?
It was a dawning realisation for me. There was a posting on ProMed Mail, that peer-sharing scientific site, that I think had flagged it very early in January. We were already talking about it, but over days it became clear that it was more than your average disease that passes from animals to humans. It was that cluster of serious pneumonia cases with clear potential to be passing between humans. So it had that SARS feel about it, which makes your heart sink for sure.
Jeanette, has it been hard in the past to explain what your work is? I think it's pretty self-evident now, but before 2020, How did you explain that to people?
So as someone said to me this morning, I'm not sure I want to be remembered like this, I'm the face of disasters in Queensland. So if there's a cyclone or a flood or a fire or contaminated water or lead problems, I'm the person who's been out there for the last 15 years. So it wasn't that difficult to then take on this increased role to really get the messages out about the pandemic.
So, Jeanette, you're the longest serving Chief Health Officer in Australia, if I've got that right, 15 years. Did you do more public announcements in the first 15 years or in the past 15 months?
Oh no, it's been the past year. I mean, day after day, 7 days a week, getting out there, getting those messages. Of course, it's been more than any other time in my career. Thank goodness, because I wouldn't want to do this non-stop, as we've had to do. But it's been critical. It's been really, really important. And I did bring part of it on myself in that I didn't want other spokespeople in Queensland because it was such a complicated message. And my view was that even if you had subtle changes in that message, it would confuse people even more. So that's why I ended up having to do so much media, and that was actually my own fault.
And then also you've been fronting the media, both of you, so consistently. Has that attention followed you? Are people like walking up to you on the street and stuff now? I know everyone knows that you've been on bedspreads. Like, what's up with that, honestly?
Yeah, look, I'm not buying them. My family's not buying them, thankfully. It's a crazy world in many respects. We shouldn't be surprised by it. We've seen public health figures globally thrown into the spotlight. That puts them in a good light, it puts them in a bad light, and the very pointed attention brings the best and worst out of people in many respects. To the extent that amplifies a good public health message, I'm very happy to carry it. But it's a crazy world. I'm very happy to do selfies. I'm very pleased to see those public expressions of support that I get walking down the street. I'm also cognizant that people have been hurt through this pandemic, that the decisions that I've made, that others have made, have implications for people's financial and employment situation and how their business is doing. And I understand the hurt and anger that sits behind people's perception of me as well. I'll carry the good with the bad because it's all part and parcel of that difficult decision making.
What about you, Jeanette? Do you have a memorable experience with a member of the public?
Lots and lots. They've been fantastic, absolutely fantastic. And it is really lovely that people come up and they talk to me. And yes, as with Brett, I've made some decisions that I know have caused enormous problems for people, and people knew that. So they came up to me and said, look, what you had to do with aged care was really hard, that they couldn't be visitors, but you've saved lives, and we're so glad you were prepared to make that decision, and they didn't need to, so they could support those families and work with them.
So as Chief Health Officers, you're talking heads to many people, but you're also human beings. Jeanette, how did you take care of yourself during what must have been like the most hectic year of your life, surely?
There were a lot of advantages because I went through the last pandemic and I sat down with my husband and said, we're not doing it that way again.
SARS?
That was awful. No, that was
Swine flu.
H1N1 swine flu in 2009, which was just a warm-up act compared to what we've been facing the last 12 months, but it was quite bad enough. So we made a few decisions at home. Our kids were older, which made it so much easier. And I started exercising. I'm not a great exerciser, but I took my own advice, 30 minutes every morning, and it's made an enormous difference.
And Brett, you spent a lot of time in lockdown. What were some of the things you really missed as a human being?
Oh, human contact. It was, it was a collective trauma for Victoria, not having the kind of interactions that as human beings we all want to have. So missing my mum, missing my brother, who obviously lived separately. I had my family at home. That was the key thing for me. But, the social interactions that are part and parcel of normal life were not available to us in Victoria for some weeks. It was a really, really long haul. I guess I had the advantage that I could go in for those press conferences. I was meeting with people for that purpose alone. At times I would have rather been at home with my family on many of those occasions than fronting up for work. And my family were my absolute rock through that period. but it was one of the toughest times of my life, no questions.
So coming sort of to looking behind to looking forward, Jeanette, what do you think this coming year looks like?
Well, this is the year of the vaccine. I absolutely believe that we've got to encourage and insist that close to 100% of our adults get vaccinated as soon as possible. That's critical. And we'll probably need to have another dose after we've had the first 2 doses to manage the new variants that are developing, but that's no problem. That'll work out beautifully because they've already developed these vaccines in an amazing time frame, which has been so good. They'll then modify those vaccines. So this is going to be the year of the vaccine.
Yeah, absolutely the same. We have a pathway out of this. It's not perfect. There won't be a perfect vaccine out there, but the alternative we've seen play out in other countries, and we should be so thankful that we're coming from a place of zero community transmission to be able to get really high-level, broad and effective coverage of our population to protect them and to change the dynamics of transmission so that if we get cases, it won't sweep through in the way that we've seen in other places in the world. That's a real privilege for Australia, and it is our pathway out of here.
Brett Sutton, Jeanette Young, thank you so much for sharing your experiences with us.
Delightful.
Thank you.
Thank you.
Well, welcome. I'm Norman Swann, and nice to see a COVID-safe audience here. Nice to see an audience at all at the World Science Festival, and I thought most of you would stay home because of the announcements today. So it's important to take the pulse of the pandemic. You've heard from 2 of our more prominent chief health officers, particularly Brett Sutton, who's been to hell and back. And we're going to have a great discussion for you and also hear from a couple of people from overseas. Now, if you're on Twitter, you can ask questions via Twitter, and the hashtag is AskWSFB. That's Ask or WSFB, and I'll try and get to your questions towards the end of the conversation. We should have plenty of time for that. It's a bit awkward to take your questions from the floor because there's no microphones, but I might do that anyway and try and repeat your questions for the recording. Let's meet our panelists. Online from Sydney is Professor Raina McIntyre. Raina is Professor of Global Biosecurity at the Kirby Institute in the University of New South Wales. She leads a research program in the control and prevention of infectious diseases. She's a vaccinologist herself. She has over 400 peer-reviewed publications, received many awards, including the National Immunisation Award, and has been very intensively published during the COVID pandemic. Please welcome Raina. Our next guest comes to you from New York. Wendy Zuckerman is a science journalist. She's worked with us at the ABC, started her career at New Scientist magazine. She created a podcast, Science Versus, and in 2020, Science VS won the 2020 Kavli Gold Prize for coronavirus reporting. That's like a Pulitzer for science journalism. Please welcome Wendy Zuckerman. And we've got full sound, so if you boo, cheer, clap, they can hear you. Don't hesitate to do any of the above. Just keep going. And finally, we actually do have somebody here live on the stage with us at QPAC, Professor of Virology at the University of Queensland, Paul Young, who has been working for many years overseas and here, developing improved diagnostics as well as vaccines for viral pathogens of both human and animal origin. Please welcome Paul Young.
Good morning, everyone.
And Paul was one of the leaders of the team that developed the UQ vaccine, and we may talk a little bit about that later. I did a recorded interview as a kind of curtain raiser, but we're actually going to hear 3 excerpts from it during our conversation this afternoon. And, well, it's now this morning, but this afternoon. This morning or this afternoon. I'll stop bumbling and get on with it. And somebody actually I've spoken to many times over the years, and I made a 4-part series on pandemics in 1990, which was based on the fact that people were thinking that HIV was the first new virus to ever hit humankind. And anybody who knew anything knew it wasn't the first new virus, and it certainly wasn't going to be the last. And I made a 4-part series. And we interviewed Michael then, because Michael had elucidated possibly the cause of the Athenian plague, which was well described by Thucydides, and has had a huge interest in past pandemics, but current pandemics as well. And in November, he was appointed to Joe Biden's transition committee, COVID-19 advisory board. He's author of the best-selling book, Deadliest Enemies, Our War Against Killer Germs. He's director for the Center for Infectious Disease Research and Policy at the University of Minnesota. And I asked Michael, I started the first section of my interview with Michael Osterholm is really looking at the history of pandemics, because almost the biology is almost the least important part of a pandemic. We make pandemics ourselves. Anyway, have a listen or a watch.
Well, there's actually several things that have to come together. First of all, you have to have an infectious agent that hasn't been in the population for some period of time, meaning at least generations, so that you don't have a large group of people who are immune to the particular pathogen. The second thing you have to have is a readily good means for transmitting it. Most of the pandemic agents that we think of, the viruses in particular, were ones that were transmitted via the respiratory route, meaning that it was actually very easy to move it quickly through large populations. The third thing you had to have with regard to this is the fact that it had a way to move from continent to continent. And we learned that even back in the Middle Ages that happened. And so when you put those three together and you have that ability to cause serious disease, you now have a pandemic of concern.
And where do social and political factors fit in? Because you certainly had, in the plague of Athens, you had a siege, you had warfare, you had malnutrition. With the Black Death, you had, again, warfare, environmental disruption, which created a mutation in Yersinia pestis associated with sea travel and trade. Just play in social and political factors to pandemics, the origins of pandemics.
Well, in a sense, we've created a modern world that is by far the best mixing vessel we could imagine for a pandemic. For example, just take a look at what the next influenza pandemic could very well represent. Today, we have over 23 billion chickens on the face of the earth to feed the 8 billion people. Over half of all the body weight of birds in this world are chickens. Then you add them being the source of the avian or bird viruses, and you put them close by to pigs. And we have well over 90 million pigs in the world at any one given day to, again, feed the protein needs of the 8 billion people. Oftentimes, the chickens and the pigs are co-located in such a way that the bird viruses are inhaled by the pigs, and their lungs are very unique in that they have the receptor sites with the ability to be infected by both bird viruses and human viruses. And so when an influenza virus from a bird and a human get together in the same lung cell, you have the opportunity for a new virus to spin forward to cause a pandemic. That didn't happen even 50 to 100 years ago, that kind of agriculture, that kind of concentrated human population, animal population together. Then you throw in modern transportation. And so in a sense, we've really been grooming the whole world for centuries to become better and better at creating pandemic infectious agents, and then once they're here, moving them around the world.
And if you add one more factor in, which has been this idea has been circulating with this pandemic, is our ability to manipulate viruses in the lab and through CRISPR to change their genes a lot. And one theory, less likely, is that it escaped from the virology lab of the Institute of Virology in Wuhan, where they were doing what's called gain-of-function studies on coronavirus, maybe 2 or 3 years before the pandemic, where they were studying what it would take to turn a coronavirus into a pandemic virus.
Yeah, at this point, I can't vouch for what was or wasn't going on in the Wuhan lab. There's surely lots of speculation. This has been an issue that I've been very concerned about for some time. I served almost 6 years on the National Science Advisory Board for Biosecurity in the United States, a group that was brought together to address just this very issue. And I've been very concerned, even if it was not by intentional means, that we were manipulating infectious agents with the idea of trying to understand what they did or how they did it, without realizing that accidentally you could create that kind of pandemic agent. So this is a very serious issue. Mother Nature surely by herself can do a wonderful job of providing us with these dangerous viruses, but humans surely can add to it. What the verdict will be ultimately for the coronavirus situation is unknown, but it's something we have to be mindful of and consider that in the future, someone could possibly create a virus like this that would be a huge challenge.
How much store do you set by the story of the 2 Chinese scientists who've been let go from this level 4 lab in Canada, the national level 4 lab in Canada, who apparently who took Henipaviruses into China, and maybe other viruses as well?
You know, I hear the same things everyone else does. I don't have any magical source of information or any crystal ball. I just come back to the fact that this is a very important issue. We have to address it, and there's no perfect answer. If someone is going to do something nefarious with infectious agents, we surely could be at the risk of having a major infectious disease problem, including a potential pandemic. I think that if one goes back and looks at the 2001 anthrax attacks in the United States, I have no doubt that the individual who perpetrated that was someone from a high-security U.S. lab who had mental health issues that intentionally moved those Bacillus anthracis spores through the mail. And so we have to be mindful that this can happen.
Just before we go back to the coronavirus pandemic, what can we do to minimize the chances of that? And some people believe it may have already occurred, it's just not turned into a pandemic yet.
This is one of the challenges of modern science and the whole issue of human values. You know, we know that people do nefarious things all the time. What's more challenging now about this issue is it's gotten easier and easier for individuals to do things with infectious agents. You know, we're now doing the kinds of studies, the kinds of activities in high school microbiology labs that literally just a decade to 2 decades ago were only done in the most secure of government laboratories around the world. Now, these are not investigations or experiments that are trying to create new infectious agents or change old ones to be more infectious or more deadly. But the point of it is that the same tool today that can be used for routine science investigation can tomorrow be the same tool that could be used for something nefarious. And we don't have easy answers right now. We just don't.
Professor Michael Osterholm, speaking to us from Minnesota. Rainer, what's your comment on what Michael was saying? And I mean, we're never really going to find out. The Chinese are blocking us from finding out really what went on with the origins of this virus.
Yeah, I think there's still a lot. I agree with everything Michael said. There are a couple of other mysterious clues, one being that the virus was apparently detected retrospectively in wastewater in Spain in March 2019. And if that's correct, how on earth did it get in the wastewater back then? And why didn't it cause a large epidemic or pandemic in March rather than December? And there were other events as well that predated that supposed December origin of the pandemic. So I think it definitely arose prior to December 2019, and we need to look at all those other lines of evidence as well.
Do you think we'll ever know?
It's unlikely. I think it's unlikely that we'll know.
So if it's unlikely we'll ever know, how can we prepare for the next one?
I think we have to be open to the fact that, you know, the methods, the accessibility and the cost of dual gain-of-function research or dual-use research of concern in infectious diseases. It's so accessible. The cost has just dropped dramatically. It doesn't take a high-level lab to be able to do quite mind-boggling research. For example, a lab in Canada that was, a reasonable lab, but not, like a CDC or vector laboratory, synthetically manufactured horsepox virus, which is very, very closely related genetically to smallpox, in 2017. And that shows that it is actually possible to synthetically create smallpox without having any virus to begin with, just created in a lab. So I think the gate's well and truly open. We can't go back and close it. We just have to learn how to manage those risks. And we also need to ask the question. If you never ask the question, is this epidemic natural or unnatural, then of course you'll never identify unnatural ones. And unfortunately in public health, the way we think is that everything's natural.
And just before I move to Paul, how would you possibly control what happens in a $150,000 biology lab with somebody with a master's but an ideological bent to destroy the West, for example?
It's very hard to control, and the question really isn't so much what goes on in documented known laboratories, it's what goes on in undocumented laboratories. We've seen the proliferation of drug labs and meth labs in backyards and do-it-yourself labs, etc. Similarly, there is a proliferation of do-it-yourself biology labs all over the world, including in Australia. But the technology is available for people to set up do-it-yourself labs that are undocumented as well. That's more of an issue. I think there's reasonably good regulation of documented labs.
Paul Young, I mean, a lot of people were expecting the next pandemic to be a coronavirus. This was not unexpected. And you'd been working on MERS, had you not?
Correct.
Tell us what you've been doing.
Yeah, I guess in the last 5 years, we've probably been better prepared for a pandemic than we ever have been. And I say that because of the Ebola outbreak in 2014-15. WHO was roundly criticised for their reactive response to that particular outbreak. I mean, they did as much as they could, but there was no preparedness component to it. And they
the criticism was stronger than that.
Well,
they squibbed it.
Correct, they did. But they turned around and within a year or so were convening bodies to investigate, well, how can we deal with this better in the future? And they came up with a strategy of preparedness of a blueprint priority disease list. So, here is a range of viruses around the world that might possibly re-emerge. And they also,
but that was partly run by the person who squibbed
yeah,
the Ebola pandemic.
True, but the outcome of all of this was, I think, a better preparedness and a more connectedness between big pharma, biotech and academia and actually addressing some of these issues.
But it took an outside organisation, not WHO, to get this going, CEPI.
Absolutely. So CEPI, well, WHO could never have funded what needed to happen. CEPI was really the funding arm of that.
You should explain
concept.
That's Coalition for Epidemic Preparedness
Innovation.
Exactly.
Coalition for Epidemic Preparedness Innovations, funded through the Wellcome Trust, the World Economic Forum, the Bill and Melinda Gates Foundation, and a number of partner countries, most notably Norway, but Australia is also contributing into it. And it's a multi-billion-dollar enterprise to fund research to develop vaccines that wouldn't normally be developed by Big Pharma, and also to develop platform technologies that might be able to deal with disease X.
So a few years ago, they had this challenge. What was the challenge?
So the challenge was to, using a platform technology, be able to move an unknown viral sequence, suddenly identified, to enough clinical material to go into a phase 1 clinical trial in 16 weeks. And that was just unheard of. I think there were 30-odd applications to that particular round. We were one of them because we'd developed over the previous 10 years a platform technology that could be applied to a wide range of different virus types, including the coronaviruses. And we had been, as you say, working on MERS. So we submitted an application with one of the few academic organisations to be funded. We received 3 years of funding starting in 2019 to develop a pipeline of vaccine activities in Australia here. Collaborators in the ANU and the University of Melbourne, tied up with CSL and a number of other industry partners. And the idea was to put together as I said, a pipeline of activities that would lead us to vaccine within that 16-week period. The plan was we were developing 3 different vaccines: RSV, respiratory syncytial virus, and influenza, for which there is a vaccine. We're using them as the comparators, as it were, and we chose MERS, ironically enough, as the one to really focus on.
Middle Eastern respiratory.
So, Middle East respiratory syndrome, which is another coronavirus. And so we spent our first year developing and really knitting together that pipeline of activity. Part of our program was actually in 2021 we proposed this in our grant application. In 2020,
This is 21, isn't it?
Yes, in 2021, we proposed in 2018 that CEPI would send us the sequence of an unknown virus. And our goal would be, our
So
task
this
would
be your
be,
little test.
a little stress test, we called it. We would develop a vaccine within 16 weeks. The only problem was that stress test came a year earlier, and that was last year.
How good was your vaccine looking?
The vaccine was looking very good. It had come through phase 1 clinical trials and was shown to be safe and well tolerated. We had very little in the way of adverse side reactions. Some of the usual sore arm at the injection site, pretty much all. But we also were able to induce a really strong neutralizing antibody response in all of the age groups that we had tested. And that was an antibody response that was above what you saw in convalescent sera of patients who'd actually experienced the real disease.
So Raina talks about this a lot, that you need vaccines that are better than the natural infection.
Correct. And hopefully those that are going to impact on the viral load in the upper respiratory tract as well. So not only protect against disease in the lungs, but also transmission.
So, I mean, editorializing, I thought it was outrageous that they canned it. Because they canned it because we don't have adequate HIV testing in blood transfusion services.
Summarised very well there. But look, I think, we began the process. I mean, remember, this was a research project that got turned into developing a real vaccine.
And we should explain, you were using a part of the HIV
Yes.
to clamp down on stabilize the spike protein so that you get a uniform immune response.
One of the problems of developing subunit vaccines, which ours is, are protein-based. So this is the outer coat of the virus itself.
So to explain, it's very different from the Pfizer and Astra vaccines where they send a genetic message into the cell. You, like the Novavax vaccine, are directly injecting it into the body.
Using proteins like the flu vaccine, the papillomavirus vaccine. So these are the proteins that stimulate the immune response. And they're essentially tricking the immune system into thinking it's experiencing that viral infection. The problem with generating those particular proteins is that they are little machines. They're nano machines that drive the entry of the virus into the cell. And to do that, they actually undergo a big structural change. And when you do that, they actually lose the shape that they have on the virus surface. So they don't actually induce the right immune response. So we developed this technology, which we called the molecular clamp, which locks them in place. We like to view it as a bit of a bulldog clip to hold the protein together. And what we used, there are a number of approaches one could use to do that, and we chose the best one that we'd tested in the lab, which happened to come from an HIV human immunodeficiency viral protein called GP41. We actually engineered it to take away all the antigenic sites that we knew were there, and so that we assumed that it would probably induce an immune response, but it wouldn't cross-react. As it turned out, it did very, very slightly in the tests that are typically run these days for HIV screening. I mean, I should have given you a slide of what the tests actually show. They were literally at the borderline of activity. It was in an area of detection they called indeterminate. But that would trigger another HIV secondary test, which obviously came up negative. So the problem was that there was this potential impact on diagnostic interference in HIV after vaccination, and so the decision was made that that was too big a risk to take forward the vaccine.
Wendy, just from your observation as a fellow journalist covering this, and going back to December, January, what was it's easy to be smart in hindsight, and you've got full permission to be smart in hindsight, but what was your response at that point? What were you thinking?
In January? That was really interesting. That was our first reporting of the virus happened in January, and we basically started tackling it because a couple of months prior, we had created this fictional pandemic story for our podcast, which was we mainly focus on facts, but we, for fun, just thought, oh, what if we just pretended there was a pandemic that went on and we got it fully cited, and Anthony Fauci had time to listen to it because, he wasn't busy back then, and we were working very closely with him.
He was only running the National Institute of Infection and Allergic Diseases,
exactly,
which had lots of time to spare.
Exactly, but he was responding to our, we were like, is this infection rate realistic? Is that infection rate realistic? And he's sort of responding at ridiculous hours to a podcast. And then several months later, we start hearing whispers of the coronavirus in China. And our listeners are saying, wait a sec, is this like horrifying scenario you guys invented? Is this happening? And in my head at the time, I was like, no. We created this worst case scenario. Like, we just need to make this episode, and like, bottom line, like, we're going to calm them all down and explain it. And then so we got Anthony Fauci back on the phone, and I was like, Anthony, so like, we made this crazy scenario. That's not going to happen now, is it? And he was like, well, it might. I don't know. And I remember me and my producer just looked at each other and we're like, no, but that's not the sort of journalism we do. We're not doing scare tactics here. Come on now. Like, this is going to be like SARS, the original. It's, China's going to contain it. They're doing these dramatic measures. And he was like, Wendy, we don't know what's going to happen. And I think that was when the penny dropped for us, where we realized things actually could get quite bad. And at the time in the US press, it was still being referred to as the virus in China, as if it was going to be contained there.
I mean, you talk about we're not in the scare business. I remember doing a late line, I was presenting a late line, and I had David Baltimore on, the Nobel laureate, and people were talking about genetically engineered organisms at that point, blah, 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 and I assumed that he was just going to be very relaxed about this. So I said, David, what keeps you up at night? And he said, smallpox, which, to Raina's point. Mm-hmm. And to talk about scare, Wendy, I mean, I had Raina on, I think, second week in January, something like that, and I said at the end of the interview, you remember, I don't know if you probably remember this, Raina, I said, give me out of 10 your concern, and she said 7. And I thought, oh, that's interesting. 2 weeks later, Raina was at 9 out of 10. What changed during those 2 weeks, Raina, that made you go to 9 out of 10? It certainly scared the shit out of me.
Well, in the first 2 weeks of January, we believed it was still a small cluster that wasn't growing, and that was because they used a very restricted case definition of severe pneumonia. So it looked like there were about 46 to 50 cases and it wasn't growing, but they actually weren't counting the mild and asymptomatic cases. And of course, it had spread all over the world by then, we now know. But by later on in January, we heard about cases in different countries, so we knew it was popping up in multiple different countries, and that's what really ratcheted it up for me.
I asked, obviously America has been infamous in this pandemic. I often say that if this pandemic had hit during the Obama years when there was more international cooperation, you probably wouldn't have had a pandemic, but it hit during the Trump years, and America still leads the world in terms of cases and deaths. So I asked Michael Osterholm about the US situation, and here's what he said.
Well, I think this virus came into the United States through any number of different sources. Our borders, in a sense, were like a submarine with screen doors. It was more than open to travel from any number of ways. As we know from studies that were done of the viruses first found in the United States, it's very likely it didn't come across the Pacific in any meaningful way in terms of entering the country here, but rather actually came via China through Europe and came in on our East Coast. But there were a number of points of entry where this virus arrived. And as such, it was here long before we recognized just how much transmission had occurred in any number of different areas in the country. We really were flying blind from January well into the middle to the end of March before we really got an understanding of just how much SARS-CoV-2 activity was in the country.
Just take us through
how you got to the situation you got to with an appalling pandemic, with an appalling and scary number of deaths?
Well, I think first of all, you have to acknowledge the fact that this disease got politicized very early. It was an issue of where we had a federal government telling the public everything was okay, we had it under control, and there wasn't a sense of urgency. I can speak to that firsthand because our center, and I specifically, put out a document on January 20th of 2000 saying this was going to be the next pandemic. Wake up, America, and get ready to prepare for this. That was met pretty much with a thundering deafness. No one really wanted to hear about it. In fact, more often than not, I just got criticism. I wrote an op-ed piece in the February 22nd issue of the New York Times last year in which I repeated not only the call for America to understand the shortcomings that we weren't ready for this at all, but here we had the World Health Organization that still did not declare it as a pandemic till March 11th, still almost another 3 weeks later. And so I think the world was really asleep at the switch as to what this pandemic could be or what it would be. And then once that happened, we just got further and further behind. And I think there was this part of America that also said, you know, we're independent souls. We're going to do what we want to do. And we had a very, very poor response. Now, I have to acknowledge that, you know, we may have given up, you might say, as a public in doing the things that could help reduce the transmission. And meanwhile, other countries in the world, such as in Asia and even in Europe, after their initial problems really clamped down. But look today, particularly in Europe. Europe is really no different than the US overall at where they're at now. The only part of the world that has continued to sustain what I would consider important control of this infection has been those countries in Asia and of course in New Zealand and Australia, where we see it's not just authoritarian governments, but democracies can also control this. Now people will argue, well, Australia and New Zealand are islands. Well, they had many introductions of the virus, and that could just as easily have taken off, and it didn't. So I think that one of the lessons learned was that if you, from the very beginning, had a plan how you were going to attack this virus, its early identification, the contact tracing, the quarantine, the isolation, and the willingness to do that, you could virtually control this virus infection. I just look no further than New Zealand and Minnesota, my home state. We have roughly the same size population. Minnesota has had almost 6,500 deaths. New Zealand has had 25. Now, is their science that much better? Not really, but their will to do something about it was. And I think that's a lesson learned, not just for this pandemic, but pandemics in the future too.
And just going back to that original discussion we had about the origins of pandemics and the multifactorial nature of them, I mean, America didn't do that well with the 1918 pandemic either. And isn't this sort of individualism that doesn't want to take community action, that individual freedom trumps, to use an unfortunate term, community action?
I think that's surely a big part of it. Also, I fear that there's a certain arrogance by us in this country that because we are the United States of America, we can't be impacted by an infectious agent like this. There was this assumption that we'll handle it. We know we have the most modern medical system in the world, or so we declare. You know, we think of ourselves as having the ultimate public health community. You know, the Centers for Disease Control and Prevention, the National Institutes of Health, worldwide recognized experts in these diseases. But what we didn't have was a real understanding of what this virus could do and would do in our communities. And as such, we weren't prepared. Our hospitals were not prepared. We were overrun with cases. We weren't prepared to deal with the social and racial inequality issues that furthered the transmission of this virus because of the kind of living conditions that many of these populations had to live in, crowding and a lack of access to health care. So I think that as a whole, this pandemic really shined a light on all the deficiencies of our country from what I would call both a humanitarian standpoint and a strategic standpoint of public health response. And we need to learn from that, surely. We are going to see it. I've been predicting that since January with the B117, and that is definitely happening. We're seeing it unfold right here in our own state of Minnesota right now.
When you look forward to the next 12 months, you've got new variants emerging almost spontaneously around the world. Not necessarily, some of them are spreading, but some of them are just occurring, and very similar mutations because of the environmental pressure of immunocompromised people. Even partial immunization is the other issue that's going on. But you have got vaccination unfolding. What do you predict, given your predictions have been pretty accurate in the past, globally for this pandemic over the next 12 months?
Well, let me just say I have one disclosure I really need to make for this presentation, and that is I know less about this virus today, I think, than I did 6 months ago. So let me, with some real humility, tell you what I think very well will happen or might happen. Number one, we've been dealing with this now really for almost 14 months as a world population, this pandemic. But we've only been dealing with the variants for 3 months. We went along for many months assuming that these variants or these mutated viruses had no real significance as mutations, just the fact that we could identify them as being different. It was only really in early November we really understood that, wait a minute, these variants have other properties that are really significant. One, some of them are able to cause much more severe disease. Number two is they're also much more infectious. Or number three is that they actually can evade the immune protection from vaccine or from natural infection immunity. And once we realized that and we labeled these variants of concern, we began to understand this was a whole new ballgame, and that the variants were going to drive this pandemic in a way we had not appreciated. I just mentioned what's happening here in Minnesota right now. Well, it's beginning to happen around the whole entire United States, and that is what happened in Europe starting in November, December with B117, the UK variant, that is both much more infectious and causes more serious illness. And even though we have seen this great reduction in cases in the United States since January, and people feeling like, aha, we're over the hump, we're just on our way to vaccine, in fact, that's not the case at all. We're now about to watch a B117 surge occur in this country that could be quite devastating, even with the number of people who have been vaccinated and the number of people who have already been infected and have some protection. Now, fortunately, B117 doesn't evade the immune protection of the vaccine or previous infection, but just the sheer transmission issues are huge. So we're going to see another wave of illness here as we go into what will obviously be the months of the summer where we will have much more vaccine. But now you ask me where we're going with this? I don't know, because one of the things that we're confronted with are these other variants, the P1, for example, from Brazil, or the P1135 from South Africa, where there we're seeing the variants that actually can evade immune protection from vaccine and from previous infection. What's happening in Brazil right now with P1 is just devastating, both in not only the city of Manaus, which was the original location where we saw a big second wave of infection due to this new variant, which actually meant people who were previously infected got infected again. We're now seeing it in Sao Paulo. We're seeing it in Rio de Janeiro. So whatever happens with these variants could determine how well our vaccines or previous protection from having been infected will actually work. And guess what? We are concentrating on getting the vaccine to those in the high-income countries right now. Vaccine nationalism at its worst. And at the point right now, we may be able to vaccinate up to 20% of the low and middle income country populations over the course of the next year. Well, guess what that means? That means that the transmission of the virus is going to continue full throttle in those countries. That's where the new variants are going to spring forth from. And when they do, they very well could challenge how well our vaccines in the high-income countries work. So I would like to think that's not going to happen, but I have to say at this point, everything points to the fact that we are going to expect to see these new variants come forward. So we have to be very, very leery of assuming just because we have the vaccines we do now that we're done, we're over it. We may not be at all.
So before we explore those latter points, which are really calm and comforting, I'm sure, to you all, Wendy, what was it like living in New York during this, amazing pandemic, which really was a failure of politics?
It was awful. It continues to be awful. I think it's almost been kind of useful. That photo suggests, there's light and people would try to bring joy to each other. But it's very scary. At the worst, at the peak of deaths, there was thousands of deaths every day. We saw this all around the world at certain times. I think it was 7 o'clock and everyone would sort of go out their window and clap for the healthcare workers, but also just to remind each other that we were around. At the beginning of the pandemic, when surfaces were thought to be when we just didn't know whether surfaces were going to be a real problem, it meant going to the supermarket was just a minefield, and bleaching all of our groceries and that sort of thing. And I think sort of seeing America, but as Michael pointed out, it's not just America fail at this. I think it's been kind of good for Australia because it has made it very easy to say this is why we need to close our borders when we have 2 cases in Queensland. We just can't sit on this because it is to live amongst it is a real to avoid using a swear word, it's a real head problem. So I think it's been tough. But I will say though that I feel like Michael is a little bit of a Debbie Downer here, and I think like when he's talking about, and I'd love to hear Paul's take on it, but when he's talking about the variants that are evading the immune system, it makes it sound like these new variants have basically made our vaccines useless. And that's not the case at all. There are no studies out there right now showing that any of the vaccines are useless. The worst numbers we have...
No, I'll have to stop you there. Study in South Africa showed that Astra was useless against the South African virus.
It did not show it was useless. It showed that there was a 60%, the lowest numbers we're seeing is 60% efficacy. That's like the flu vaccine.
No. There was a study showing they looked specifically at the South African variant, and in fact there was no effect, but they kindly said 10% effect. So there's a study just published in the New England Journal of Medicine. Do you want to expand on that, Raina? You've got your mute on.
Classic.
Yeah, in the combined study that was published in the Lancet, the second trial, they showed it was about 20%, but the New England Journal study, which was just patients from South Africa, when they looked at the efficacy against the South African strain, it was 10%, but it crossed 0. So,
for all kinds of disease, or just for serious, or just for mild?
Against the B117 against the South African variant.
So what do you think, Paul, about this variant issue? Because this is the one, so we've rolled the dice on Astra. Mm-hmm. And Astra is pretty good at preventing severe disease, probably as good as Pfizer at doing that. But it's not that good with variants. I think the South African variant anyway, and maybe the Brazilian one, that's yet to be seen.
Yeah, I think maybe just to wind back a bit and just talk about variants and how they emerge. I mean, it almost sounds like the virus is thinking about how it can avoid things. Viruses, particularly this sort, just mutate randomly. It's just part of their makeup. They actually have a built-in error code so that they can actually develop a population of viruses that have different potentials to emerge depending on the selection pressure. So they're just randomly mutating, and then various selection pressures will allow ones to pop out. Now, one of the selection pressures for this virus is it's spilt over from a bat population, so it's not perfectly adapted to humans. And so some of the adaptation that we're seeing in these mutants is not against immune pressure, it's actually binding more effectively to the receptor in humans. Various mutations in that binding site that are occur randomly are being selected for in the population, and they're growing up. And that's an outcome we didn't really expect. I mean,
but essentially, it's an evolutionary pressure in response initially to lockdown.
Correct. Yeah, but most virologists, I mean, certainly I teach my students that as a virus co-evolves with its host, it tends towards less virulency, because what the best outcome for the viral population is a population, a host population that can transmit it efficiently. And if you kill your host, that's not going to happen. But in this case, we've got that rare situation where the virus is being transmitted in part by asymptomatic carriers as well as symptomatic individuals, particularly the younger individuals. And so the death that it's causing in the more susceptible age groups and the higher age groups are not impacting the viral population as a whole. So what we're finding is that the adaptation to better binding and therefore more replication in the respiratory tract is the one that's being chosen. That's causing increased disease. So we are seeing an adaptation to virulency. Now, in terms of the vaccines, they had for a while. I mean, I was asked this question last year, and I was saying, look, you know, all the evidence would suggest that these random mutations that are occurring and are starting to adapt are still protected by these vaccines. But we are now seeing the emergence.
And that seems to be emerging from people who are immune compromised, so people who are on cancer therapy. And it's not any surprise that they're emerging in South Africa and Brazil, where there's a lot of people with HIV not terribly well treated. What's going on in somebody who's immunocompromised that creates a potentially vaccine escape mutant to survive?
Well, my view on that is that there is the selection for these viruses that are growing more efficiently. So there's replication more efficiently. The antibody response, the immune response that is most protective, is actually binding the same site. Most of the strong protective antibodies bind to the site on the virus that it uses to bind to its receptor. And in the absence of those, you're getting this replication, viral replication at higher levels in these immunocompromised people and in large populations that is almost irrelevant in the context of the immune pressure that's there. But once the immune pressure comes along as well, then you get that secondary component, and I think that's how they're coming up. You're getting this dual host adaptation and immune escape.
So Ryan, take us, I mean, do you think that Michael's scaring us the way Wendy suggests?
Look, I think it's sort of, I think both Michael, I agree with Wendy that there is quite a bit of hope with the vaccines, and there's more development now into different ways of overcoming this issue of variants of concern arising, including booster shots, but also more universal methods that'll, target the more ubiquitous proteins or parts of the structures of the virus to generate an immune response, no matter what the variant is. So I think that will be, a challenge that can be overcome. The problem is, though, that some of these variants, particularly the B117, which isn't so badly affected by the vaccine, it's slightly reduced, but not too badly, it's much more transmissible and much more serious. So some people are saying it's like another pandemic, a new pandemic. And I guess the one we really have to watch out for is B1, the South African variant. Maybe the Brazilian variant. We haven't seen as much data on that. Maybe Paul has a better idea than I do, but I think there's been the other two variants have been better studied. And of course, there's other variants popping up in Japan, in Los Angeles, in New York, etc. We know which the actual mutations are that are concerning. We just have to see. I would expect to see this year that one of those strains will become dominant. At this stage, it looks like it's the UK variant B117 that's kind of taking over everywhere. And if you remember back to a year ago, the original strain that arose in Wuhan kind of died out by March and was replaced by a different strain that became dominant around the world and was responsible for most of the epidemic activity in the US and Europe last year. And similarly, I would expect these new variants to become dominant this year.
So go on.
I was just going to say, I mean, one obvious solution to this, or one approach to this, is a bit like what we do with influenza, and that is we're going to be developing new vaccines based on the technologies that have now been shown to work, but with the new variants. And we may be looking at vaccines in a wave 2 approach, wave 2 of vaccines, that is, a second generation of vaccines that are multivalent. So the combination of the dominant variants at the time.
Multivalent meaning different versions of the same virus, like they do in influenza. And this universal vaccine that Raina was talking about, people have been, this is like the
Holy Grail.
The Holy Grail of influenza vaccine. Can you have one vaccine that just lasts forever and ever? And failed miserably at it with influenza. Didn't succeed with HPV. We've had to have multivalent vaccines for HPV. Is there any chance?
I think that is research that's going on in the background. We're doing similar work on what we're hoping is a universal flu vaccine as well. But you're right, we haven't had much success in that regard. We know what the targets are on the protein. It's actually developing an approach where we can force the immune system to target that particular area. We can do the same for SARS, and I know that is happening in the background. But I think the easiest, lowest hanging fruit is going for a vaccine that contains components of each of the different variants.
Wendy, I want to talk to you about politics for a moment. I mean, arguably the New York epidemic was caused by Cuomo, because he sat there for 3 weeks knowing that there was a pandemic, said that he was almost imitating Trump by saying our hospitals are the best in the world, we're able to cope with this, and didn't take any public health measures. And then he became a hero. Dan Andrews was the health minister in 2009 in Victoria when they ran up the white flag in Victoria and stopped doing testing because they'd run down their public health resources to this, from a great state of public health to poor, and yet was the hero in Victoria. What's your observation? I mean, you're not essentially a political journalist, but what's your observation of the politics of this and the pandemic?
The life of a white man, I suppose you've just given us. I think as Michael mentioned, and as you've said, the politics of New York and America widely is a mess. But I don't think we can say that Cuomo started or is responsible for the pandemic in New York. I mean, New York is when you look at all of the factors, how dense it is, how you have different multi-generational families living, grandma's taking care of the kid, and the adult of the parent is going to work, say they're an essential worker, bringing that to these home that's going to affect the grandparents. You have this like hotbed. And I think it's when we think of the really high density places around the world that manage to control this disease, and I'm thinking China, basically, like Wuhan. When we spoke to people in Wuhan at the height of their lockdown about what was actually happening over there in order to clamp down on the virus, I mean, the infrastructure was unbelievable. So, for example, in some apartment buildings, people would not go shopping, like grocery shopping on their own. Instead, it was organized that a community member there would be a WeChat for the entire, these huge apartment buildings. It's not some 20 people, it could be hundreds. We would have a WeChat where they would say, this is what we need for the day, for the week. Then one member would go to the market, purchase it, wearing a hazmat suit, would come back, and then one by one, the families would come and get the food. And to imagine that sort of thing happening in New York it just wasn't there.
The point that I'm making here is that he waited, and in those 3 weeks, with an R0 that's well above 1, he got an exponential growth, and there would have been far fewer cases in New York. I mean, vastly fewer, and easier to control via lockdown, and he didn't.
Obviously, acting early is the key, but I hate to be a mouthpiece for Cuomo, but at that time, it was so difficult to know what the numbers were and what the right thing was to do. Now we're all so clever, we're wearing our masks and everything. But at the time, I remember talking to Anthony Fauci about the first case where it was one of the first cases of known coronavirus was in Chicago. And then it was a secondary infection to their partner, basically. And I remember calling up Fauci and saying, is this a problem? We've got our first secondary infection. He's like, it's just this. We think maybe it's okay. You know, even Fauci was like, we're not sure which way it's going to go. So to lock down all of New York before we knew what was going on. And remember, Norman, America didn't even have proper testing until so long. So we didn't even have a handle on the cases. So to act faster, yes, obviously would have been amazing. But I do have some sensitivity for what it would have been like in those rooms to know exactly what to do at the early stages of the pandemic.
Yeah, I would take issue with Michael's comment that the world wasn't prepared. The reality was, certainly in the space we were working, you know, we decided to do our stress test vaccine production in early January after Christmas, hearing about the Wuhan outbreak. We basically had a lab meeting and said, you know, when the sequence comes out, we'll put this into the stress test. But literally by the time that sequence came out on January the 11th and we began our study, CEPI contacted us and said, we think this is real, and we're going to probably call on you to go down the full path of vaccine manufacture. And within 2 weeks after that, they'd asked us officially to do that. So, you know, there were around the world people who realized in early January that we were on the cusp of something that was going to be quite serious.
So, Ryan, to give us the balance here between, I mean, when something like this hits, and we'll come to the next pandemic in a minute, but when something like this hits, you don't have any tools apart from the non-drug ones, the non-vaccine ones.
Yeah, and I think that's where many countries were unprepared, in that we all saw what happened in China. Like, they locked down, I think it was January the 23rd. We could see the epidemic curve. It was a classic epidemic curve, which showed a very rapid upswing of cases, an exponential rise in cases over a very short period of time, a peak, and then a decline after that lockdown. So I think other countries, you know, in Europe, in the US, etc., they did see what happened in China and that non-pharmaceutical measures were used and worked. But I think the issue there was probably a combination of culture and leadership. So I think someone mentioned earlier, cultural factors are really important. Public health measures, non-pharmaceutical measures, are essentially draconian in nature because you have to stop people having contact with each other to control a respiratory transmitted virus. And that is much less palatable in highly individualistic cultures. When you've got the deadly combination of individualistic culture and bad leadership, that's when things can really come unraveled. And that's exactly what we saw in the UK, in the US, in other countries in Europe, where the leadership wasn't able to, you know, compensate for the cultural reluctance to take up non-pharmaceutical measures. And that includes the masks, the social distancing, sometimes lockdowns, etc. All those things work. I think there was also a loss of corporate knowledge in expert committees around in many Western countries. Probably they had experts in sort of basic science, clinical medicine, etc., but not enough public health experts who understood that things like contact tracing and quarantine work, that testing is the most important measure. You have to identify all the cases and isolate them to stop them infecting other people. And then you have to identify their contacts and quarantine them because they're going to be the next tranche of people who get infected. Those two measures alone can control an epidemic or a pandemic in various settings. But the testing issue, as Wendy mentioned, it's still not, you know, they're still not on top of it in the US. And one of the issues there was, well, they never got across the testing issue. And, you know, many countries switched to that kind of, you know, they have the Defence Production Act, which the current US government has activated to pivot manufacturing to support medical and healthcare needs, but that didn't happen last year, and so we saw massive shortages of masks and PPE. You know, I had healthcare workers from America contacting me saying, I can only get a cloth mask. Should I be treating COVID patients wearing a cloth mask? And that's a really hard question to answer. I mean, my ideal answer is you shouldn't be working unless you're adequately protected. If you're a miner and you're asked to go down a mine shaft without proper protective equipment, the company will face litigation and prosecution. But unfortunately, we allowed our healthcare workers to be like cannon fodder and did not protect them adequately, still don't protect them adequately. So I'm sort of wandering off on a few different tangents there, but in any pandemic, before you've got vaccines or drugs, the non-pharmaceutical measures are the only way you can control it. They are effective. But I think that there wasn't that understanding of how important those measures were.
And in a transition to think about the next pandemic, I mean, if you look at our committee structure, we've no idea what the principal committee here for health, HPCC, how they make their decisions. We've no idea how the infection control expert group make the decisions. They still have in the national guidelines that you don't need to wear a P2 mask when you're looking after a patient. No wonder the medical registrar at Princess Alexandra, which is the cause, that she caught it from a hotel quarantine patient, and then this cluster we're seeing now, because we followed the national guidelines in Queensland and she wasn't wearing an N95 mask. And we don't, the ATAGI, the Australian Technical Advisory Group on Immunisation, we don't know how they make the deliberations. So we now have issued a very weak advice on the thromboembolism problem with Astra, whereas the British Society of Haematology, the German Society, has issued very specific guidance for doctors there. Are we too secretive, Paul?
Yes, I think we are. I mean, there's been a lot of debate in infectious disease circles about the need for, in Australia, a CDC, an open and robust Centre for Disease Control.
That didn't help America during Trump.
I was just going to say, I don't think that
I was going to
work.
say, Paul, what open and transparency?
Yeah, no, I said an open one.
No, but America, the American committees are fully transparent.
Yeah.
When they make their decisions, you can see how they make them, you can examine them, you can interrogate them for all the problems they've had.
But I think a combination model of what we have now, I think we have some state health departments that have really run the show quite well around the country. What we need to have is interconnectedness and common protocols. So I think...
But what's all this secrecy? Why can't we find out why they make their decisions? Raina?
Well, I don't know, but even the UK, the SAGE committee there, all their minutes are public. Yep. You can look them up on the web and read every, bad decision that was made and pick it to pieces. But I think transparency is important for public trust. If you want to bring people along with you on different policy decisions, it's really important that there's transparency on how decisions are made.
I think you've got furious agreement on that one.
Yeah. Let's just look at the, cheery though this conversation has been, the next pandemic, which is really the last question I asked Michael Osterholm is, when and where does he think the next pandemic is going to occur?
I don't know. And I think that's part of the challenge we have today. Remember the last influenza pandemic in 2009 originated in Mexico after everyone assumed it would start in Asia. We're maybe not as surprised that we had the coronavirus pandemic start in China because, in fact, the concern had been there for some time about coronaviruses in animal populations in Asia. But, tomorrow it could start anywhere in the world for influenza. And another coronavirus could surely pop up, just like in the Middle East, where MERS, Middle Eastern Respiratory Syndrome, suddenly popped up in 2012. So I think it really is a global surveillance we need. Any day, anywhere, any place, it could happen. Our job is going to be early recognition and doing the most we can to try to contain it if possible, and if not, what else are we going to do than to try to protect the world?
Michael Osterholm, thank you very much.
Thank you very much.
So Wendy Zuckerman, have you got an appetite for talking about the next pandemic, or are you still...
Still too Debbie Downery. No, I think this has taught us, of course, that pandemics happen. I mean, it happened in 1918. It's happened, as we've mentioned many examples, and there will be another one. And I think, we started the conversation kind of talking about the more nefarious ways that a pandemic can possibly spread. But I think it's important for people to know that, like, in history, the pandemics that we have all happened, naturally. And the vast majority of scientists in this space do believe that this one also happened naturally. And just because we haven't found the exact bat that has the coronavirus, that doesn't mean that anything nefarious happened. So, but I think, the modern world we live in is just ripe for another pandemic, and probably it'll happen before 100 years, I think.
Raina, what needs to be in place for us to be well prepared for the next one?
So I think we do pretty well, as Paul said, with the pointy end of things, with drugs, vaccines, etc. But we could do better in early detection of epidemics. And, when you think about the pandemic arising in Wuhan and those first cases, if that had been detected before it had spread outside of China, it could have been stopped. The pandemic could have been prevented, right? If it had been detected early enough to stop it with the measures like isolating cases, contact tracing and quarantine. So I think we could do a lot more. And there's actually a huge amount of open source data out there, social media, news reports, etc., that is uncurated but vast, and it can be tapped to obtain epidemic signals. Now, police and law enforcement people do this to monitor for signals of terrorist attacks. You can do the same thing for outbreaks because people talk about illness. News agencies report on it long before an official notification lands on a health department desk. And we've done a whole bunch of research on this showing that you could pick up signals even for Ebola in 2014. We published a paper in PLOS One, I think, where we showed you could have picked it up on social media, looking for combinations of words and using certain algorithms 3 months before WHO knew anything about it. So I think, and we did another study searching on Weibo in Chinese for signals of the coronavirus pandemic prior to December, and there was a signal that something unusual was happening in the middle of November. So I think we could do a lot better. Some countries use this kind of epidemic intelligence. So Malaysia, for example, has a system called EpiWabak where they scan social media and news reports, etc., and then they send a team out. If there's a signal, they send a team out to investigate it, to do the testing, etc. Obviously, you will get false positives, but to pick up an early signal, you can't just rely on official notifications. Even clinical syndromic surveillance, which health people tend to think is the absolute cutting edge, is quite delayed. So I think we could be doing better in that space.
So Paul, tell us how your vaccine research is going now.
Yeah, so we went through a stage gate.
Thinking of the next pandemic.
Of course. We went through the stage gate with CEPI, so a determination of whether they would continue funding us, given that we weren't going on to phase 2, 3. We had a very detailed dive into the results that came out, and they were very encouraged by the positive data we had got out of the phase 1 trial, so have funded us further, and indeed have asked us to stay focused on COVID. So we're looking at generating a new clamp that's got nothing to do with HIV. We've chosen some candidates and they're looking good, and we're focused on looking at variants. So that's the COVID path. But this technology does work for a wide range of those viruses that are thought may emerge, and generic ones, another coronavirus, another influenza, and so on. We do have that technology to apply. So it's about ramping it up so that we've got that pipeline of activity now we know works. Last year proved that we could do this in a year and develop that vaccine approach. But it's still a year to 18 months for developing a vaccine. I think the new messenger RNA technologies will probably allow us to to kick into play with the vaccine approach rather earlier. We've proven that, at least in Australia, public health measures work or can work. So we've got some of those tools. I agree with Michael that surveillance is the key. Improving our surveillance of potential spillover events into the future is where we need to go in some regard. But there's an untapped resource that we also need to explore, and that's the area of antivirals, generic antivirals. When you make a vaccine, it's just the nature of viruses. The exterior protein tends to change between viruses. So we make a SARS-CoV-2 vaccine. It's not going to work against SARS-CoV, the original one. It's not going to work necessarily against the next coronavirus that comes out. But what all of that particular family of viruses share are a common strategy for replicating, the way they grow inside cells. And they have their own enzymes to do that. And enzymes, to retain that functionality, have to be fairly common. So if you target an antiviral, develop an antiviral to one of those enzymes, it's going to work against another coronavirus as well, or likely to. And I just take my mind back to SARS, because some of the research that followed the SARS outbreak in 2003 was to do exactly that, develop drugs that would work against the polymerase and the protease, two key enzymes in the replication of those viruses. And they actually developed drugs that worked in the lab, but there was no commercial imperative to take them forward. SARS had disappeared. I think we're in a changed environment now, at least I hope.
Well, I mean, if I take us back to last March, people were saying, oh, it's going to be impossible to produce an effective respiratory vaccine. Don't hold out any hope that they're actually going to be successful, because they haven't been that successful in the past. Ryan used to pick me up on that, because claiming that measles is actually a respiratory virus, and we're pretty good at that. However, people said, well, the breakthrough is actually going to be treatment. And there hasn't been one. I mean, there was dexamethasone, which is 20% reduction in mortality. That's not bad. But there hasn't really been an effect. Remdesivir is just a waste of time and money, isn't it?
Correct. These are all drugs that are being repurposed. They're already existing drugs that have been licensed for use in humans, and we just wanted to see whether they would work also on corona. What I'm talking about are drugs that are designed specifically for coronaviruses. And so what you do is you generate one of these drugs that will work well. Testing it in phase 3 trials is going to be challenging, but getting it to the point where it can be in the toolbox next time another coronavirus comes out. I was also one of the ones saying last year that actually it's going to be relatively easy to make this vaccine. Everyone was picking up on the fact that we'd not made a coronavirus vaccine in the past. Well, there was a good reason for that. There was no commercial imperative. SARS disappeared. So the good vaccines that had been developed, potentially developed, hadn't been progressed.
We've got 3 or 4 minutes left. Let's see what questions are up on Twitter for the panel. Yes, why don't you shout out, sorry, hold on a second. Why don't you shout out your question, and I'll take one from Twitter. Yep.
Okay, I'm quite interested in the fact that all of this is post-factual, so the pandemic's already started. The evidence is that it's really environmental destruction, such as forest destruction, wetland destruction. We happen to used to live in Laos, where our local wet market had 87 zoonotic diseases, actually just in one market. So, I mean, I'm just wondering...
So I hear what you're saying. I'll ask Ryan. So the question is one of the core causes here, and which will perpetuate environmental disruption, environmental change, encroaching on new environments, the way you sell fresh food in wet markets. Ryan?
Yes, that's definitely an issue in the increase in emerging infections we've seen over the last decade. There actually have been more and more serious emerging infections arising in the last decade than any time previously in history, and changes in the way we live and farm and produce food have all contributed to that.
A question here, under what conditions will the World Health Organization declare the pandemic over? Well, they were late to the party coming into it. Are we going to be early going out of it? What is the way out of a pandemic?
I don't really know the answer to that question. In terms of a sharp endpoint, I mean, you're quite right. I mean, many people were calling it before the WHO officially called it a pandemic. But there is an expectation. I mean, there's been a debate for some months now. Are we going to eradicate this virus, or will it become endemic in the human population? I think I'm leaning towards the latter now. And
did you ever think it was eliminatable?
Yeah, I did actually.
Did you?
Well, it's unlike influenza. It looked like it was a tractable target, and as long as we got global reach of our vaccines, and I'm starting to doubt that we're going to get equitable access to vaccines throughout the world, then that would have dampened it to a point where it couldn't have transmitted any further. So there was a potential of eradication.
Rana, what's your answer to the way out?
So eradication means the virus does not exist in the world. And the conditions for an eradicable virus are, firstly, that it only has a human host. When there's an animal host, it becomes much more difficult, maybe impossible. And secondly, that there's not much asymptomatic transmission. So SARS-CoV-2 has both those things, a likely animal host and quite major asymptomatic transmission, which is going to make it impossible, I think, to eradicate. Elimination, however, I think is possible. So countries or regions can eliminate the virus by using very high efficacy vaccines and achieving really high coverage. My dog thinks so.
I hope your dog doesn't have a coronavirus, otherwise it's going to be spreading it everywhere, Raina. Well, look, that's all we've got time for. I'll take one more question from the audience. There you go. So this is a common question that we get on CoronaCast. If somebody's wanting to get pregnant, should they have the vaccine or not? By the way, before we move on, if influenza vaccine is a must, there's no doubt about the fact that you must have that. Paul, from looking at the evidence,
There isn't a lot of evidence. The trials haven't been done. I mean, the best guess would be that that's not going to be a huge problem, but there isn't that cohort in clinical trial studies that were really investigated.
Quite a lot of pregnant women have had it in the United States, haven't they, Raina?
Yeah, I think it was, go Ren, sorry.
You go, Wendy, go on.
Oh, I was going to say, I think it's it's a question I also get asked quite a lot, and I think it's a risk-benefit analysis, given that we don't have the studies. So I think in Australia, that risk-benefit changes a lot versus, say, if you're in New York. So I think probably the biggest risk is that sometimes in the second dose of the vaccine, some people get fevers, and sort of fevers aren't considered to be a good thing when you're pregnant. And so if the caseload is really low and your baseline likelihood of getting coronavirus is really low, you might want to wait until the pregnancy is over. But having said that, there have been many women who have gotten the vaccine both in China and in America now, and I think all around the world. Unfortunately, we just don't have the data to be able to really say the outcomes just yet.
Raina?
Yeah, I agree with everything Wendy said, and I think in the US, people are being given a choice, so they're giving informed consent to get vaccinated, understanding that there's no clinical trial data. And absolutely, it's a question of situational risk. So if you're in a country with with serious epidemic activity, it's probably better to get vaccinated. But if you're in a country without any community transmission, then I think it's better to wait.
Could you please join me? We're a fantastic panel, and we've all learned a lot. So please thank Paul Young, Raina McIntyre, Wendy Zuckerman. And thank you to all you Queenslanders for joining us here on this day of another cluster evolving in Queensland. Let's hope the cluster is now terminated. I hope so, because I'll never get back home, and as a Mexican. But thank you again for coming, and enjoy the rest of the festival. Thank you.